Hi, Assalamualaikum. We continue to the last subchapter in chapter 13. Okay, plant response to stimuli. Okay, light cues many key events in plant growth and development. Cues here meaning light serve as a signal towards many key events in the plant growth. So light reception is important for measuring the passage of days and season. Okay, so light also function to determine the days and season for plants. And plants not only detect the presence of light, but it also detect the direction, the intensity and the wavelength color in the light. As you know, the sunlight consists of many colors radiation, so plants can detect uh, certain radiation in light. So it can be categorized into phototropism, photomorphogenesis, and photoperiodism. Okay, what is photomorphogenesis? The effect of light on plant morphology. Plant morphology meaning the characteristic, the external characteristic of the plant. For example, the structure of the leaf, the bending of the stem, the movement of the root, or the structure of the root. Okay, the external structure of the plant we call morphology. So when there is effect of light on plant morphology, we call as photomorphogenesis. From the light, it causes the morphogenesis. For example, developmental changes including germination, flowering, and fruiting. So in plant, in order to detect the presence of light, plant need some protein. This protein we call as phytochrome. Okay, so phytochrome is a protein which attach to a non-protein component, which is the chromophore. Okay, so chromophore is function as a light absorbing pigment. Okay, so the pigment that absorb light is the chromophore, and this chromophore attached to a protein called phytochrome. Phytochrome is the photoreceptor responsible for opposing effect of red and far red light. So in phytochrome, okay, actually plant have two types of photoreceptor. The very popular receptor, only two. The first one is blue light receptor. The second is phytochrome, which is function to absorb red and far red light. Okay, so in this chapter we just discuss on the phytochrome. So this phytochrome, they have opposing effect of red and far red light. Later we will see what is the meaning by opposing effect of red and far red light. So phytochrome consists of two identical protein. This one, two set, okay, that form to form a functional molecule, and each of these protein has two domain. The first one is for photoreceptor activity, which function as the photoreceptor covalently bonded to a non-protein pigment, which is chromophore. And the second domain is the protein kinase activity here. Okay, so this photoreceptor domain interact with the kinase domain to link light reception to cellular response triggered by the kinase. So kinase is an enzyme. So this kinase enzyme is function to perform activity. Okay, activity in plant. For example, the produce of enzymatic enzymes. Okay, or the transpiration process. Okay, so all these process, all the biological process that happen in plants is conducted by certain enzymes. So this is the photomorphogenesis. This is the PR and the PFR. Okay, PR represent the chromophore that absorb red light. Okay, so PR only can absorb red light radiation, and PFR is the chromophore that can only absorb far red light. Okay, so the one more thing that you have to know, PR is the inactive form of the chromophore, and PFR is the active form of the chromophore. So when this PR is exposed to the red light, they will convert to the PFR, which is active form, and later they will form the responses such as seed germination, or the flowering process, or the fruiting process. Okay, and if the PFR, so it is reversible, and when the PFR is absorbed far red light, it will change or it transform into the PR, which is inactive form. What happened during this, as I mentioned previously, the PR is inactive form, so that the plant become inactive in all their activity. So the enzymatic destruction will be started, and any developmental will be slow. Okay, no more development when the PFR convert to PR. So this is some description that you can read. Phytochrome exists in two photoreversible states with conversion to PR to PFR, which trigger many developmental responses. So this including seed germination, flowering, fruiting. Okay, and the red light triggers the conversion PR to PFR. Okay, PR only can absorb red light. Okay, so the red light will convert the PR to PFR, which is the active form. Meanwhile, the far red light will trigger the conversion of PFR to PR. Okay, PR is the inactive form of the chromophore. The conversion of PFR is faster. Okay, PR convert to PFR is faster compared to PFR convert to PR. Okay, so this is logic, because plant need to do developmental and plant need to grow. So the conversion from PR to PFR must be faster. Okay, sunlight increase the ratio of PFR to PR. So PFR is needed more, okay, compared to PR, because PFR will induce growth, okay, will induce development. Okay, that is the importance for the plant, right? Okay, so now we go into biological clock and circadian rhythm. Okay, what is biological clock and what is circadian rhythm? Okay, so these two referring to the body clock of plant, and biological clock and circadian rhythm is not in plant, but in all eukaryotes and even human we have these two. For example, the most common is the sleep wake. The sleep wake, okay, that happen in human. We have a proper time, what time we sleep and what time we will wake up. Okay, so that is the biological clock and circadian rhythm. Same goes with the plant. Many plant processes, for example transpiration, the production of some enzymes, okay, certain enzymes to conduct developmental and growth in plant. So these type of processes, they oscillate during the day. Oscillate mean they are keep changing, they are not stagnant. Okay, some plant need more time, more night to flower. Sometimes they require more sunlight to flower or to germinate. So different plants or different species, the requirement are also different. So this cyclic response to environmental stimuli we call as circadian rhythm. Okay, and the circadian rhythm, okay, it takes 24 hours. Okay, it takes 24 hours for a species to be happen, and the clock may depend on synthesis of a protein regulated through the feedback control and may be common to all eukaryotes. So from this slide, it means that every species, they have a specific requirement in order for them to grow and to develop. Okay, so example, legumes. Okay, so many legumes, they lower their leaf in the evening and raise them in the morning. So this is one of the routine for legumes plant. They will lower their leaf in the evening and raise them in the morning every day. The same routine will be happen for this type of plant. So we can call this is one type of the biological clock and circadian rhythm for this plant. Okay, so now we go into the effect of light on biological clock. Okay, so light represent many cues, or it can be signal, okay, serve as a signal to many developmental process in plant. Okay, if we can see here, phytochrome, okay, phytochrome, which is the element, okay, the element in plant that is function to induce growth or to inhibit growth in plant. So it plays a major role, okay, in plant development. Okay, so the phytochrome conversion, okay, phytochrome conversion refer to the PR to PFR or PFR to PR. So that is the meaning by phytochrome conversion. So this conversion will mark the sunrise and sunset. Why it said mark the sunrise and sunset? Because during sunrise, okay, the red light is more compared to the far red light, but during sunset, the far red light, okay, is more compared to red light. So this phytochrome conversion, when the phytochrome conversion from PR to PFR, we know that during that time is the daytime, because the chromophore changed to the PFR, more red light. When the sunset, we know there is a sunset, the plant know that is the sunset time, because the PFR changed to the PR. Okay, so that is the meaning by phytochrome conversion mark sunrise and sunset. So this will provide biological clock with environmental cues to the plant. So plant will know the change in time by having interchange in the phytochrome conversion. So now we go into photoperiodism. What is photoperiod? Photo means light, okay, period means time. So it relates to the length of the night and day. So this photoperiod is the environmental stimulus that plants use most often to detect the time of the year. Okay, so photoperiodism is a physiological response toward the photoperiod. So these two are terms, okay, just understand the terms. Okay, now we go into photoperiodism and flowering. Okay, how the duration of night and day will give influence to the flowering. Okay, some developmental process, including flowering in many species, they require certain photoperiod. Okay, based on the amount of day length control, flowering plant can be classified into 4 groups. So according to the duration of night and day, these plants, okay, they are grouped into 4. The first one is short day plants. Okay, in short day plants, meaning this plant require very long night, okay, in order to flower. So this group of plant, they need more darkness compared to day so that they can flower. So this plant normally flower during late summer, during fall or winter. Okay, the second group is long day plants, also known as short night plants, meaning this type of plant, they require very short night compared to day. Okay, flower when the light period is longer than a critical number of hours. So this plant normally found in the late spring or early summer. The third group is intermediate day plants. They flower when night length is intermediate length. Okay, so neutral, 50-50, equal. Okay, for the fourth group is day neutral plants. So this type of plant, they are unaffected, not affected by the photoperiod. They will flower at certain stage of maturity. When they reach maturity, regardless of the day length at the time, they will flower. So that is the day neutral plants. So these are the experiment, okay, that has been conducted in 1940s. These researchers, they want to discover that flowering and other response toward photoperiod. Is photoperiod will stimulate flowering? So what they found is the flowering is actually controlled by the night length but not the day length. So the main point that control the flowering is the night length. So this is the experiment. Okay, if you can see in short day plant first. Okay, so the first image, okay, when the day length is longer compared to the night, there is no flower. Okay, when the day is shorter compared to the night, flowering. Okay, when there is day is shortened compared to the night, but there is interruption here, they give a flash of light, no flowering. Okay, also no flowering. Now we go into the second experiment for long day or short night plant. So this plant, they require very short of darkness but more day length. So the first, they flower, of course, because of the day length is longer. And the second, the day long is shorter compared to darkness, no flowering. And the third is the day length is shorter but there is interruption of light here, but it is flower. Why? Okay, so we will see on the conclusion. Okay, the experiment indicated that flowering of each species was determined by a critical period of darkness for that species, not by specific period of light. Therefore, short day plants are more properly called long night plants, and long day plants are really short night plants. Okay, so that is the conclusion. But from the experiment, you can see that there is interruption of light here, but in this plant it still flower. Okay, why? Because the duration, even though they introduce a flashlight, okay, interruption of light here, the duration of light, if we compare to the darkness is still longer compared to the daytime. That's why this plant still flowering. Okay, so now we go into action spectra. Action spectra refer to the radiation, okay, the color of the sunlight. Okay, so our sunlight, they carry many radiation. There is blue, green light, okay, red light. Okay, so this action spectra referring to the color in the sunlight. And the photoreversibility experiment. Okay, from the word photoreversibility, you must remember the red light and far red light previously. Okay, so red light can convert into far red light convert into red light. Okay, so this show that phytochrome is the pigment that receives the red light, which can interrupt the nighttime portion of the photoperiod. So this experiment, a unique characteristic of phytochrome is reversibility. So the experiment is used, okay, is conduct to test the reversibility character of the phytochromes in response to red and far red light. To test whether phytochrome is the pigment measuring interruption of dark period, researchers observe how flash of red light and far red light affected flowering in short day and long day plants. Okay, let's see. The first plant, okay, the short day plant or long night plant. Okay, so it requires a very long night, but here the night is shorter compared to day. That's why no flowering. Okay, and for this one, the second row, it flower because the night is longer. Okay, the third row, no flowering because, okay, even though the night is longer, but they introduce a red light here. If you can see, when the presence of red light, okay, what happened? It will interrupt the dark period. Okay, it will interrupt the dark period. Okay, let's see number 4. Okay, row number 4. The plant is flowering. Okay, here it introduced red light and far red light. It's still flowering. Why? Because when red light followed by far red light, the far red light, okay, far red light will cancel the presence of the red light. So far red light will cancel the presence of red light, like there is no interruption at all. So that is why this plant is flowering. Okay, but for the row number 5, it shows that when red light introduced to far red light and then introduced back to red light, so it means the darkness is interrupted by red light. So there is no flowering here. So what you have to know or to understand, the far red light will cancel the presence or the action of red light. Okay, when the flash is ended with far red light, it seems that no interruption of light at all. That's why this plant is flowering. Okay, just read several times on this experiment in order to understand the functions of the red light and far red light in flowering. So the conclusion, a flash of red light shorten the dark period. Okay, when there is a flash of red light here, it shorten and it bother the dark period that the plant needed, because this plant is a long night plant, so they require a very long night in order to flowering. When there is interruption here, okay, it cause the dark period become shortened. That's why the plant is not flowering. Okay, a subsequent flash, of far red light cancel the red light effect. That's why in the 4th row it flower, because this far red light cancel the action of the red light. Seems there is no interruption of light. Okay, there is no interruption of light. And same goes with the 5th and the 6th row. Okay, so this reversibility indicated that phytochrome that measure the interruption of dark period.